Hello everyone, my name is Naima Ben Sheikh and I am the lab supervisor. Welcome to the clinical biochemistry laboratory at the Charnikol University Hospital in Tunis, Tunisia. Through this door, this is the specimen reception area where we register and blood collection before they are sent for analysis. This is the first and the most important steps in the laboratory process. First, we enter the patient information into the system. When we prepare the appropriate tubes, we specify barcode for each test. These tubes are then used for sample collection from the patient.
Next, we collect the blood samples using sterile equipment while following hygiene and safety rules. It's important not to choose the correct tubes and to follow the correct order of draw to avoid the contamination between additives in the tubes.
Hello, this is Asia. We will talk about stock management. This is reagent and consumable storage room. We will here keep reagent chemical tapis. Here we keep reagent chemicals tapis, gloves, syringes, and other consumables used every day in different sections of the laboratory.
Over here is the main biochemistry session where all the tests are performed and processed here. Every sample has a specific barcode. The barcode tells the system where the tube needs to go and in which analyzer needs to be treated. First, all samples need to be centrifuged in those machines. After that, they go to one of the analyzers. Different analyzers perform different tests. In total, we have 4 chemistry analyzers in the lab.
Let's start with the core lab. This is the Actelica solutions that we use as our main analyzer. The system processes many different types of samples. It's flexible and automated, that performing a high number of tests. First, we have the sample handler. It has 16 positions dedicated to controls and calibrators. The samples move through the system using the Maglite transport system. Then they go to the clinical chemistry module, which performs many routine chemical tests. This analyzer uses microvolume technology, so it only needs about 50 microliters of sample and can perform up to 15 tests at the same time. This is the immunoassay analyzer. It uses disposable types and has a second incubation rings, which allows us to have a fast result. For example, the troponin result can be obtained only in 10 minutes. Every day we perform quality control check to make sure the tests are accurate, and we select the controls for each test in the analyzer. Sometimes we also need to perform calibration if the test stability period has passed. The system can recognize them and run the correct tests, and the final results are validated by residents and sent to the other departments.
Here we have the capillary 3-octa. This is a fully automated analyzer that can perform several tests and helps improve the speed and efficiency of the laboratory. For quality control, we simply load the QC materials onto a special rack and place it in the instrument. The system automatically reads the RFID code and barcodes and runs the controls in each capillary. When we are ready, we run patient samples, we load drugs with primary tubes onto the instrument. Different types of samples can be loaded at the same times. After the samples are processed, we review the results in the forage system. We first check the quality control results in the 8 capillaries, and only after that we validate the patient results. The system uses capillary electrophoresis, which is mainly used for diagnosing conditions such as multiple myeloma, hemoglobinopathies, and diabetes mellitus. It helps the laboratory work more efficiently while providing valuable clinical information.